mutants with enhanced activity. So why did we focus our attention on Cas12a, especially considering that SP-Cas9, arguably the most common CRISPR enzyme, has such good on-target activity like we're showing in this table here? Well, as several people have alluded to all day, SP-Cas9 does have good on-target activity, but it can have some problems with specificity. And that puts a lot of studies in danger for off-target effects and unintended editing, which is obviously a huge problem for therapeutics. And that's where Cas12a can be a preferred choice since it has intrinsically high specificity. We also find that particularly the LB variant has high temperature tolerance, which makes it a great choice for zebrafish and plant genome engineering. And also the patent space for Cas12a proteins is a little bit more open. FTO is a little bit nicer, and that can lead to easier discovery. So we focused our attention on the Acidomonococcus and Lactnospiraceae variants of Cas12a. And our goal here was to make a Cas12a that is as potent and reliable as Cas9, but also maintain the specificity and all the attributes that make Cas12a an attractive choice to begin with. So we started with directed evolution. We set up a cleavage reporter system. So a very simple system here with a high copy plasmid in E. coli. And that plasmid has two very important elements, one being a target site for either a canonical triple TC PAM or the less efficiently cleaved triple TT. And then the second part of that plasmid contains a toxin that when produced will kill the E. coli cell. So the only way to get survival through this system is to cleave this plasmid. And that's just because bacteria typically rapidly degrade linearized DNA. So we're asking Cas12a to cleave that plasmid before the toxin is induced. So in the lower half of this figure, I'm showing the performance of wild type in this system. So without the toxin, this plate shows all of the colonies that could have survived. And the plus toxin shows everything that survived by cleaving the triple TC. So you can see that cleaving the canonical PAM, a lot of clones survive, whereas on this other side, the triple TT, very few colonies make it through this screen. So from here, we did low fidelity PCR to generate a mutant library. We did several rounds of this. We put it all through this screen, collected the surviving mutants from the triple TT plus toxin plate, and subjected everything to NGS. And here we found kind of three mutation hotspots through the Cas12a open reading frame. And from there, it just took a little bit more tinkering at site 1 and site 2 to finalize what would be our ultimate high activity enzyme. And that's how we identified and developed what we're calling the AsCas12a mutant ultra protein. So here I'm showing the performance of the ultra protein in a direct comparison to the wild type protein. And this was about 100 sites in Jurkat cells and HEC293 cells, targeting all around the human genome. So clearly at the triple TV, so that's anything with A, C, or G in the last position of the PAM, the average rate of editing using the ultra mutant is greatly improved. You can see most of these come up to 99, almost 100% edited. And since what we were looking for was indeed the ability to cleave the quad Ts, we see that come up a little bit with the ultra mutant as well. But we were very pleased with the performance of the ultra mutant across all PAMs. So for this reason, we generally still recommend that people use triple TV when they have the choice, but these triple TTs do become available with the ultra mutant. And of course, we were worried that our screening method would lead to loss of that attractive specificity that Cas12a has, or that it would change the PAM binding. So we made sure to put AsCas12a Ultra through some DNA binding assays. This is a protocol called SpecSeq, where we directly compared the DNA binding affinity of the Ultra mutant versus the wild type. And here in a plotted direct comparison, you can see that the relative binding of Ultra is much higher with DNA binding than the wild type enzyme. So we were very pleased to see that. And not only did it maintain the binding affinity, but we also find that the binding specificity is very much identical between the Ultra and wild type enzymes. So that was good news to us, as we had definitely kept an eye on literature mutants of Cas12a that reported having broadened PAM and enhanced activity. But when we tested those ourselves, we found that the reason they had an enhanced activity was because of a decrease in specificity. So we found that with our Ultra mutant, we were able to increase editing activity, but leave the specificity and binding affinity alone. So not only did we improve the performance over the wild type Cas12a enzyme, but we found that the editing levels we were achieving with the Ultra protein were pretty closely matched with what we see with SpCas9. And obviously we can't compare exact site for site, since the PAMs here are wildly different, but we do see very close matched levels of editing in matched genomic regions. And for that reason, we feel pretty confident that we met our goal here, that we provided an increase in activity for the Cas12a mutant that made it as potent as Cas9, but didn't change the specificity at all. So here we feel pretty confident recommending Cas12a for all genome editing projects, not just when Cas9 isn't available, not just when you're in an AT-rich region, but Cas12a can be used for any genome editing project. And here we see the utility of a potent enzyme like the Ultra mutant. This is HDR with a 6-base insert on an SSODN across 20 different sites in the human genome at a range of editing levels. And this is just showing that really good HDR rates can be achieved with Cas12a, despite what's in the literature or lack thereof, that we find Cas12a to be a really great tool for HDR as well. And I mentioned that we are also working on an LB mutant. This one isn't as finalized as our AS variant, but this started by just looking at the sequence and structure of both proteins and aligning them. And we did find that the AS and LB variants are very closely matched for structure and homology. And so we originally had thought that we would just port over the mutants from the AS Ultra and make an LB Ultra, and that worked fine, but we are still doing mutations to see what would be the best combination of mutations for LB. But what we did identify with our initial LB Ultra mutants is that not only did we improve the activity of the Ultra, of the Cas12a protein, we also found that the increased activity made it easier to be temperature permissive. So here at 30 degrees, we see increased activity with the Ultra mutants, and that was true for LB and AS Ultra mutants. And of course, we're still, this is a work in progress, so just wanted to flash up a little bit of how we compare single mutations or stacked mutations to identify what is going to be our final LB mutant. So just to sum up, Cas12a is very desirable as a genome editing tool because of its high specificity, temperature tolerance, and great FTL. And through bacterial screening and directed evolution, we were able to make a version of AsCas12a that rivals Cas9 for editing efficiency and potency. And since we were able to maintain binding specificity of the wild type enzyme, this could be a great choice for therapeutic applications, and also has utility for mediating high rates of HDR. And lastly, the AS Cas12a variant is available on our website currently for purchase, but the LB Cas12a is still a work in progress. It's still in development, but we are hoping that it will still have the increased editing efficiency, better temperature tolerance, and broad utility in plant editing. And we're open to beta testers for LB Cas12a if anyone finds that interesting. Last, I just want to thank everyone at IDT CRISPR team and encourage everyone to visit our website. We have a lot of good protocols on there, good explanation of our product line, and some good resources for papers and relevant material. And I'm happy to take any questions. Now you can always email me directly, or please stop by my poster at the session tomorrow. Thank you.
Okay, it's a great talk. I think we have about two minutes for questions. We have a couple in the Q&A right now. So first we see, from your studies, which Cas12a member would you recommend which can't tolerate even a single nucleotide mismatch in the guide RNA? Actually means in the target sequence. So which is the most stringent?
Yeah, is that question asking between AS or LB? I would probably recommend our AS Cas12a Ultra for its higher activity. It's slightly higher activity than the LB variant. As far as which can tolerate a mismatch best, I'm not sure we've ever looked at that.
Okay. All right, we have another question that's got some thumbs up. Do you design your HDR experiments differently to account for the overhangs left by Cas12a?
No, we do not. We have spent about 2 years developing Cas12a HDR to get it to the levels we have now, but we've never really, we don't design with overhangs. We don't really take that into consideration. We just assume that there's going to be chewback of that 5' overhang anyway, so it doesn't really factor that much in HDR.
Great, and we have about 1 minute left. So I'm just going to ask, so there's an HDR design tool that I've used on IDT for Cas9. Is there any potential of there being a Cas12a HDR design tool?
Absolutely, yes. We are working on a version 2 of that HDR design tool, which will incorporate Cas12a.
Excellent, looking forward to it.
Great. Thank you.
Okay. Hello, everyone. My name is Kenta Yamamoto, and I'm a researcher in the lab of Dr. Brandon Moriarty and Dr. Bill Weber at the University of Minnesota. And I'll be talking about Cas9 and AAV-mediated integration of chimeric antigen receptors optimized for natural killer cells for the treatment of advanced stage ovarian cancers. And I would also like to take this moment to thank the organizers for coordinating another successful year of the Genome Writers Guild annual conference. So in the recent decades, there have been incredible advances in the field of cancer immunotherapy, which involves taking the patient's own cells, the patient's own immune cells, in order to combat cancers. And through these, there have been remarkable improvements in the outcomes of a variety of cancer types. So probably the most well-utilized examples of this is the alpha beta T cells, which makes a part of the adaptive arm of the immune system. However, more recent findings have suggested that immune cells from the innate arm of the immune system, such as natural killer cells or NK cells, may offer additional advantages in the context of designing immunotherapies. So NK cells offer a multitude of unique advantages, first and foremost being their natural cytotoxic ability. So NK cells express a wide array of native and non-clonal receptors, which are able to detect and target cancer cells in order to allow for cytotoxicity. And in terms of the clinical context, NK cells have garnered a lot of interest as well for their allogeneic potential, being that they have minimal risk for causing graft versus host disease. And furthermore, in terms of safety, due to the relatively short lifespan of NK cells in vivo, the thought is that they have lower risk for causing chronic or long-term toxicities for patients. And also it's been suggested that NK cells pose a lower risk for causing cytokine release syndrome compared to conventional T cell therapies. However, the effectiveness of NK cells can be greatly hampered by tumor immune escape mechanisms. So this includes factors such as the downregulation of stress-induced ligands such as MYC-A and MYC-B, which normally would be detected by NK cells and targeted for eradication. And furthermore, immunosuppressive factors such as TGF-beta within the tumor microenvironment have been shown to downregulate key NK receptors such as NKG2D or CD16, as well as hamper their production of cytokines. And even in the case that NK cells are able to overcome these obstacles, with prolonged and persistent stimulation, it's been shown that NK cells can become functionally exhausted, which poses yet another challenge for this approach. So one method for augmenting the activity of NK cells is to express a chimeric antigen receptor, or CAR, at their cell surface. So a CAR utilizes a single chain variable fragment, which is essentially your antigen recognition domain, which is going to specifically target peptides or antigens overexpressed by cancers. And then a CAR also employs a CD3 zeta signaling domain, as well as in more recent generations, a CD28 and or a 41BB co-stimulatory domain to allow for even more activation of lymphocytes. However, most studies that use CAR NK cells, including those in the clinic, have used CARs that have been developed and optimized for the activation of T cells and not NK cells. So to circumvent this limitation, our lab previously generated and developed a novel CAR containing a transmembrane domain derived from the NKG2D NK cell receptor, as well as a co-stimulatory domain derived from 2B4, which this too is natively expressed on NK cells. And this NK-optimized CAR showed improved activation and cytotoxicity when expressed at the surface of natural killer cells. And given these results, we were encouraged and wanted to apply our finding in order to develop a therapy for advanced stage ovarian cancers. So ovarian cancer is the most lethal gynecologic disease with a less than 30% 5-year survival rate for epithelial ovarian cancer. And a big factor in this is that many patients are diagnosed at an advanced stage, which further contributes to the poor prognosis. And given that mesothelin is highly expressed in approximately 70% of ovarian cancer cases, we wanted to combine our optimized NK CAR architecture with an anti-mesothelin single chain variable fragment in order to retarget NK activity towards ovarian cancers. So to do this, we first started by using a lentiviral transduction approach in order to semi-randomly integrate our CARs into the NK cell genome. So to do this, we essentially isolate peripheral blood NK cells from PBMCs and then expand them via co-culture with genetically modified feeder cell lines. And once NK cells are thoroughly activated and proliferative, we're then able to transduce these cells with lentivirus, and in our case, lentivirus encoding either a conventional T cell CAR or our NK optimized CAR. So one obstacle that we faced early on in this work is that historically, NK cells are very inefficient at viral gene transfer, including with lentivirus. So in order to find a solution to this problem, we designed our constructs so that each one of these CARs were followed by a 2A self-cleaving peptide, followed by an RQR8 molecule. So RQR8 contains a CD34 epitope that is very specifically and strongly recognized by a monoclonal antibody that is widely used in the clinic. And being that the CAR and the RQR8 molecule are co-expressed, we're able to label the RQR8 in order to immunomagnetically select for CAR-expressing NK cells. So this is just a flow cytometry panel showing RQR8 expression, but we see that upon initial transduction with lentivirus, we're showing very low levels of CAR and RQR8 expression, in this case about 8.5%. However, after 2 rounds of RQR8-based immunomagnetic selection, we were able to achieve an average of about 80% expression of our CAR and RQR8. So with these enriched CAR NK populations, we then wanted to functionally validate their activity against ovarian cancers. So in order to do this, we co-cultured our CAR NK cells with either a mesothelin low expressing cell line or a mesothelin high expressing ovarian cancer cell line. And I'll also point out that what we're looking at here is the expression level of CD107A, which is a marker for NK cell degranulation, as well as interferon gamma, which is a pro-inflammatory cytokine. So in our co-cultures with the mesothelin high expressors, we see that compared to the conventional T cell CAR, our NK optimized CAR is showing a significantly higher level of CD107A expression, indicating greater degranulation, as well as greater levels of pro-inflammatory cytokine production. And another thing I'll point out here is that regardless of the CAR architecture that we're employing, our CAR NK cells showed minimal reactivity towards mesothelin low expressors, indicating that our CARs are operating in a highly antigen-specific manner. And encouraged by these results, we then sought to perform site-specific integration of the CARs into the AAVS1 site of NK cells, this time using a homology-directed repair-based approach. So in this method, we're electroporating NK cells with Cas9 mRNA, as well as guide RNA specific to the AAVS1 safe harbor locus. So this allows the generation of a double-strand break at the targeted site. And soon after the electroporation, we're transducing these cells with recombinant adeno-associated virus serotype 6, or AAV6, which encode our CAR2A RQR8 cargos, and also includes homology arms complementary to the AAVS1 site. So by using our AAV6 as a repair template, we're able to precisely integrate our CAR and RQR8 cargo into the genome of NK cells. So in terms of the overall workflow, it's actually quite similar to the lentiviral work that I showed several slides ago, where upon activation of NK cells, we simply electroporate them with Cas9 mRNA, AAVS1 guide RNA, and then subsequently transduce them with the AAV encoding the CARs. And again, after site-specific integration is achieved, we're performing immunomagnetic selection in order to enrich for populations of interest. And this again shows similar data of RQRA expression. However, compared to our lentiviral data, upon initial, the initial integration level is much higher when we use this Cas9 and AAV-based approach. And given this high initial level of integration, after just one round of RQR8 selection, we were able to achieve well over 90% expression of CAR and RQR8. So this again is showing RQR8 expression. And for both the T cell CAR shown in red, as well as the NK optimized CAR shown in blue, upon initial transduction, we're seeing a little bit shy of 40% average expression, but upon immunomagnetic selection, we're consistently able to achieve over 90% expression of CAR and RQR8. So to conclude, our NK-optimized CAR containing NKG2D and 2B4 domains induce greater activation as well as degranulation of NK cells compared to conventional T cell-based CARs. Furthermore, CARs can be used to retarget NK cells to mesothelin, which is overexpressed in ovarian cancer cells. We show that this occurs at a high rate of specificity. We further show that NK cells are highly amenable to site-specific integration of CARs using this homology-directed repair-based approach. And finally, RQR8-based selection allows for facile and effective enrichment of co-expressed cargo, which allows us to achieve a high level of expression of CAR and primary peripheral blood NK cells. And degranulation and intracellular cytokine assays are ongoing for the CAR NK cells that were edited using Cas9 and AAV. And additional cytotoxicity assays, as well as in vivo studies, will be conducted using these same cells. And I'd like to thank the principal investigators of our lab, Dr. Brandon Moriarty and Dr. Bill Weber. And I'd also like to thank the co-authors of this project, Emily Pomeroy, Walker Lauer, Knut Lauer-Harry, and Molly Wang. And this work definitely would not have been possible without their many contributions. So again, I'd like to give a big thanks to these individuals. And finally, I'd like to thank the members of the Moriarty and Weber labs, as well as their funding sources at the National Cancer Institute, the Randy Shaver Cancer Research and Community Fund, and the Children's Cancer Research Fund here in Minnesota. And with that, thank you for listening, and I'd be glad to take any questions either now or in the breakout sessions that will be hosted in the coming minutes. Thank you.
Great talk. I don't want to have to run through questions, so there's one in the chat if you just want to record it. And so let's maybe meet up in the breakout sessions for any specific questions speakers, and we can see all you in the next session.